Hello, everyone. Great to see so many old friends, new friends. Thank you to Frank and to Michael for organizing this really well put together conference. Great to follow Eric's brilliant talk as well. So I'd like to bum you out with a discussion first of cancer. There have been some advancements which have been big in the news in terms of checkpoint inhibitors, et cetera. But despite this, metastatic cancer is still mostly fatal. It kills 10 million people per year, and that's 40% of everyone we love is going to get cancer. But sepsis is actually even more deadly, kills more people than all forms of cancer combined, 11 million dead every year. And many who die from sepsis had otherwise survivable conditions. And there have been over 100 clinical trials in sepsis, all of which have failed. So clearly, we need new therapeutics, but we also need new therapeutic concepts. And that's what I'm going to talk to you about today. Specifically, rejuvenation via depletion of pathogenic soluble factors by nanots. So I'm Lou Hawthorne, the founder of the company, but I'm also the inventor of the technology that I'm going to show you today. So nanots essentially invert the drug paradigm of the last 2,000 years. Rather than adding molecules to the body, nanots deplete molecules from blood that drive or enable disease. They're fast, they're safe, and they're very disruptive. So let's go back in time to 2005. That was when a very important paper was published called Rejuvenation of Aged Progenitor Cells by Exposure to a Young Systemic Environment. But most of you probably know it as the heterochronic parabiosis study. To review, heterochronic parabiosis involves stitching together the circulatory systems of two mice, an old one and a young one. And I'll show two pairs just to clarify what's going on. So the idea is that blood from the old mouse goes into the young mouse and induces an aging of the younger parabiont. And that's attributable to progerotic factors in the blood of the old animal. At the same time, blood from the younger animal goes into the older animal, and it induces a rejuvenation of the older parabiont, which is attributed to antigeronic factors or some magic element in the young blood. And this was dogma from 2005 until 2020. Starting in 2020, there were a series of publications that came out from the Conboys again, and also Dobry Kiprov, who's one of the world's foremost experts in apheresis, who's here today presenting in the 4:30 slot. Don't miss it. And this latest study is called Old Plasma Dilution Reduces Human Biological Age. And the idea here is that this new work confirms part of the hypothesis of heterochronic parabiosis, which is that there are indeed progeronic factors in the blood of old animals. But it largely disproves the second part of the hypothesis, that there's something magical in young blood. And that's because dilution with saline plus albumin led to almost all the benefits previously attributed to young plasma. The best thing about TPE, or therapeutic plasma exchange, is that it's legal when overseen by a physician. This is me getting TPE in the clinic of Dobry Kiprov. I have a special type of plasma of TPE that I have done in which my plasma is exchanged with the plasma of about a dozen of these adorable little puppies. Don't worry, though, because I buy them in bulk to save money. Of course I'm kidding. Of course I'm kidding. That's Dobry and Regina's pet. That's Paco, and he's wonderful, and he's totally fine. There he is back at his job driving for Uber. Anyway, the takeaway from heterochronic parabiosis and TPE is that organismal rejuvenation depends more on the reduction of negative blood factors than addition of positive factors. And the question is, what's most important to take out of blood? There's so many things in there. There's cytokines, chemokines, autoreactive antibodies, toxins, etc. We subscribe to the inflammaging branch of gerontology, which the term was first coined by Claudio Franceschi in 2000, and this is his 7 pillars. Lots of gerontologists use 7 pillars. Franceschi's pillars put inflammation at the center, and he argues that all of the pillars are shared by aging and age-related disease. And he specifically highlights the role of 3 molecules, TNF-alpha, IL-1 beta, and IL-6, all of which rise with age. They're broadly pathogenic and poorly druggable. Let's zero in on TNF-alpha. TNF-alpha is actually part of what we call the signal power couple, two very important molecules. TNF begins life on the surface of an immune cell. It's cleaved off in very close proximity to an aberrant cell. It's the key inflammatory cytokine in the body. Its target is the TNFR1 receptor on a non-immune cell, a transformed cell like a virally infected cell or early cancer. Upon ligation, it transduces a death signal into that cell and destroys it. But under certain circumstances, the TNF receptor can be cleaved off of a cell membrane into a soluble form called an sTNFR. It's no longer capable of transducing any kind of signal. It's now just a decoy or inhibitor of the TNF molecule. These two molecules are responsible for so many health and disease processes in the body, they deserve a close look. So in health, TNF kills bad cells, all of the cell types in the upper left. In disease, it still kills cells, it just kills the wrong cells, inducing the pathologies in the lower left. sTNFRs defend cells by neutralizing TNF-alpha before it can bind to membrane TNF receptors on cell surfaces. Now, in health, sTNFRs defend good cells, privileged tissue like the placenta and the fetus, and they also function as a general inflammatory sink. Now, in disease, sTNFRs do the same thing. They defend cells, they just defend the wrong cells. All solid tumor cancers surround themselves with sTNFRs to neutralize incoming TNF. We think senescent cells do the same thing, so do MRSA-infected cells. sTNFRs also drive the immune paralysis phase of sepsis and other cytokine storms. Now, TNF inhibitors are the largest drug class of all, but they're an imperfect drug class because they all reduce immune competence for reasons that our technology will not. But sTNFRs are a completely undruggable target because an antibody directed against soluble TNF receptors would also block membrane TNF receptors, and that pathway must be kept open for innate immunity in particular to function. What's really interesting about sTNFRs is that dramatic clinical data already exists on the treatment of late-stage metastatic cancer via depletion of sTNFRs by therapeutic apheresis. So therapeutic apheresis differs a bit from TPE, so I'm just going to explain that quickly. So this is the schematic of TPE. Blood is withdrawn from the body, it goes through a plasma separator, the plasma is discarded. At the same time, saline plus albumin is combined with blood cells from the separator, and the reconstituted blood goes back into the patient's body. Now, therapeutic apheresis has much of the same cycle, except the plasma doesn't get discarded. It gets run through an affinity column, which specifically depletes just the target it's built to deplete. And this, in this case, at this clinic, the International Immunology Foundation in Germany, their affinity column depletes sTNFRs. This technology has documented the highest efficacy in cancer on the planet. They've treated over 100 patients with late-stage metastatic disease, essentially all terminal, representing 25 different solid tumor cancer types, and they've documented an ORR, or overall response rate, of 60%. Now, state-of-the-art oncological therapeutics, checkpoint inhibitors, small molecules, et cetera, really can't go beyond about 10 to 20%. So this is remarkable, particularly with some very difficult-to-treat cancers like metastatic prostate cancer. So depleting targets from blood can clearly rejuvenate an organism and treat disease. But is there a more scalable, less invasive way? That's what we're developing. Nanots are a new class of medicine. They deplete pathogenic molecules from blood, but they don't engage normal cell surface proteins at all. That's where the vast majority of drug side effects come from. You can think of nanots as the world's smallest, most specific, non-toxic sponges. And the way they work is there's a core, which is the structural element. It's completely biodegradable. Onto that, we attach capture agents, any molecule with specific binding affinity for the target. Next, we attach a porous shield, and this is really the secret sauce of the nanot. It makes the nanot specific to soluble targets and inert relative to all cell surfaces. And finally, we attach a stealth coating just to delay immune recognition and clearance of the nanot until the capture process is complete. It ends up being 150 nanometers across. You inject it like a drug, but it's cleared naturally by macrophages. And we build nanots using click chemistry, which won the Nobel Prize this year, and it's every bit as good as they say it is. Click chemistry is one of the reasons the Gen 2 nanots work so amazingly well. They're blazingly fast. They can deplete whatever soluble target they're programmed to deplete by greater than 95% in less than 5 minutes. And they can be programmed to suppress targets for minutes, if that's desirable, or up to about 16 hours. So we've already made nanots against the mouse version of STNFRs and tested it in 4T1, which is a mouse model of triple-negative breast cancer. It's a metastatic model, so you're looking for a delta of metastasis. Nanots blocked metastasis as a monotherapy, no other treatments involved, at 67% target depletion on our Gen 1. With the Gen 2 nanots made using click chemistry, we can deplete routinely by greater than 95%. So we're developing a whole toolkit of 8 nanots, 4 against soluble inflammatory targets and 4 against soluble inhibitory targets that drive dozens of diseases with unmet need. And this is our pipeline. So 4 nanots against inhibitory targets that we're going to test in cancer, but we think they'll also work as senolytics. I'll say more about that in a minute. And 4 against inflammatory targets that we're going to test in FDA-recognized autoimmune diseases, but we think they should also be effective in inflammaging. And the combination of anti-inflammatory nanots can be used as a toolkit for treating sepsis. No one's ever cracked sepsis, but we think they just didn't have the right tools. We're collaborating with 3 of the world's best hospitals for our first in-human studies: Mass General under Keith Flaherty, who co-founded Loxo, which sold for $8 billion in 2019, Mayo Clinic, and these 2 centers are going to be testing 2 different nanots against 2 different tumor-generated immune inhibitors. And then in sepsis, we haven't announced it yet, but we have a new collaboration with the Cleveland Clinic to test that. So let's take a minute and just talk about sepsis. Sepsis is an immune system overreaction. It can be triggered by various causes, infections, injuries, etc. It kills more people than all forms of cancer combined. There's no current FDA-approved treatment right now, and there's a growing consensus among researchers, which you see in the scientific literature, speculating that no single drug is ever going to solve sepsis. And the current treatment is just crisis management. So sepsis is mainly driven by 3 inflammatory cytokines, which rise in blood over a period of several days. And all 3 must be depleted to save most patients. It's TNF-alpha, IL-1 beta, and IL-6. And if these sound familiar, they're the same 3 inflammatory cytokines thought to drive inflammaging, which raises a really interesting question. Is sepsis vastly accelerated aging, or is aging slow-motion sepsis? So we've already made nanots against TNF-alpha and tested them in mice that receive a lethal TNF dose. This is a quick and dirty test of nanots for treating cytokine storm. We dose them with TNF, and we chase them with anti-TNF nanots, and we shift the mortality curve. This shows we can intervene in sepsis and other cytokine storms. These nanots will also be a next-generation TNF inhibitor for autoimmune disease because unlike all the TNF inhibitors on the market, they won't reduce immune competence because they don't disturb membrane TNF. So nanots for treating cancer via depletion of tumor-generated immune inhibitors may also function as senolytics. And I don't have time to go through all of this, but the idea is that senescent cells secrete all of the same immune inhibitors that cancer cells secrete to neutralize incoming attack by immune cells. And when you look at the pattern of immune inhibition generated by senescent cells, it's very, very similar to what tumors generate and what the placenta generates. And we've made nanots against 3 of these targets. We're going to make nanots against all of them eventually. So an interesting point is that tumors and senescent cells, like the fetus, must inhibit all the cytotoxic immune factors coming at them in order to survive. But immune evasion can be curtailed if just 1 or 2 of these factors is removed. So safety, obviously, is very important with a new therapeutic. The nanot safety profile is very promising. We haven't seen any toxicity, and we've dosed rodents at up to 100 times what we plan to dose in humans. So we think they're safe. We have a great IP portfolio. We invented the technology, and we have 15 granted patents with protection through 2037. These patents, there's 6 foundational US patents covering a range of designs, hundreds of targets and all the sources of those targets. And we have all the foreign counterparts needed in the countries with strong nanomedical industries that might compete with us. So I want to just end with a few thoughts on valuation. I know there's a lot of my fellow science entrepreneurs here and also some investors. We're all trying to think about how do you value a technology. So I also heard there was a contest for the most pretentious new biotech term. My submission is preclinical epistemology. Epistemology is the branch of philosophy concerned with what do we know and how can we know it. And that's really important when you talk therapeutic valuation, because the valuation of a therapeutic or platform like nanots is the full potential of that platform minus certain discounts. And the biggest category of discounts is uncertainty or risk. And that's mainly a problem in the preclinical phase because much uncertainty gets resolved in human studies. And the lower the uncertainty, the higher the defensible valuation that you can claim. Now, I'm not going to go through this whole thing, but I'll just give you an example of how we frame nanots relative to drugs for some of the standard concerns around uncertainty. So in terms of feasibility, that's always a concern with a typical drug, that it just might not work or might have unreasonable cost of goods. We've already demonstrated that these things work, and the COGS for nanots are the same as they are for a drug. Toxicity is a big concern. The materials we're using are completely safe. Our capture agents are shielded. And we already know that depletion of the targets we're going after is safe because it's been done in apheresis. Translation, there's always a concern that something that works in mice won't work in humans. Investors love to say we've cured cancer 1,000 times in mice. Well, we're not developing a pharmaceutical, we're developing a biophysical intervention, and the biophysics are the same across all species. Different species just have different volumes of blood. IP, strong drug IP is actually quite rare. Most drugs aren't novel. They're some sort of knockoff of an existing drug. But our IP is unique. Anyway, I won't go through the others other than to say that skepticism regarding drug performance in the clinical phase is warranted, and we have a pretty good idea of how nanots are going to work even now in the preclinical phase. So we have a great team. Our CTO, who does our nanot engineering and is also our IP genius, John Dodgson, is here. So if you see him, say hello. And we also have a wonderful advisory board. A couple of our advisors are here. Brian Kennedy, of course, you know him, and Robert Skalick, who's an expert in the role of inflammatory cytokines and cardiovascular disease. He's here. So thank you very much.